Thank you for giving me the opportunity to share my research here. I'm Anwarul Islam. I'm from Bangladesh. I have recently submitted my MPhil thesis from James Cook University, Australia. And currently I'm in Bangladesh at the moment. Today I'm going to talk about the comparison of algae, spent coffee ground, and sawdust biochar performance on silver removal. So I'm going to talk about the background of my research and the knowledge gap I identified, and the research objective and outcomes, and finally the conclusion of my study. So the background. So silver is basically a transitional heavy metal having the kind of a 107.86 atomic weight. And we see the silver like the metallic silver, silver compounds, and silver nanoparticle. And especially the silver, it has the distinct property such as antimicrobial property, high thermal and electrical conductivity, and special optical properties. So all those distinct properties make silver species useful for medical and healthcare, functional textiles, personal care products, food packaging, and electronics. And all those uses leads to the wastewater stream at some point of time. And finally, the silver residue is passed through the environment through treated effluent and also the biosolids, and which is toxic for ecosystem and also toxic for human health. Now, when the silver species come to the natural environment, the transformation we can see, oxidative dissolution happens where the oxygen rich environment. Silver nanoparticle can be transformed into silver ions. Then the sulfidation, which is one of the most stable form when the environment is hydrogen sulfide rich, then the silver species basically transform into silver sulfide. Then the chlorination, when the chlorine-rich environment, especially in contact with seawater, silver species can transform into silver chloride or silver chloride complexes. And finally, the photoreduction. It can also produce from the silver ions to silver nanoparticle or silver humic acid complex. So all those transformation reaction, it depends on the surrounding environment and the conditions, and it can be reversible. So in the natural water, we will see silver ion or silver sulfide or the silver nanoparticle. So to get rid of this toxicity from the silver, we have the current, like the conventional wastewater treatment facility, which has two segments we can see. The primary treatment, which is basically the screening and the primary clarifier, and the secondary treatment, which is the like the microbial oxidation, which help us to remove the like the pollutants. But the problem with this technology, which we are using at the moment for the silver removal, especially this system is basically bacteria dominant process, which means silver can create the toxicity for the bacteria and remove the performance of this effluent treatment system. And this system is also unable to meet the regulatory guideline when the required concentration is 0.1 to 3.2 microgram per liter. So the alternative technology which are available for silver removal, we can see sequencing batch reactor, activated slash process, ion exchange, membrane filtration, and reverse osmosis. And all those technologies has some advantages and some disadvantages. For example, like the first two processes are the bacteria dominant process, as we already said that silver can be toxic to the bacteria and remove the performance of the reduce the performance of the like the effluent treatment system. And the third and fourth, like the ion exchange and membrane filtration and reverse osmosis, of course, they are very effective to remove any sort of metal and or the pollutant. But the problem is they are very expensive for industrial scale use. And finally, if we see the adsorption, that can bring the breakthrough because the full removal is possible and it's low cost, eco-friendly, and easy operation. And maybe some of the disadvantages, maybe the process may be a little bit slower than the other process, we will see. Then for the adsorption, the available adsorbent in the market, we have so far activated carbon, clays, bio-waste material, cellulosic material, zeolites, and graphene. And all those adsorbents have some advantages and some disadvantages. So especially if we see like the activated carbon, which is the most commonly used commercial adsorbent for the water treatment and also like the air filtration system, in terms of silver removal, the working pH is 4. So the wastewater treatment plant having the silver residue, the pH is nearly 7 to 9, something like that. So which means we need to add extra chemical to bring down the pH to work with activated carbon. Then after the treatment, we need to again increase the pH, adding extra chemical to make them like ready to discharge to the water body. But when it comes to biochar, it can be produced from biomass waste. It is low cost, require less energy and less chemical compared to the other adsorbent. And it can also reduce the greenhouse gas emission, because when we are using those like the biomass waste, eventually we are reducing the load from the landfilling, where the biomasses just produce methane, which is 1,000 times more like the responsible for the greenhouse gas effect. And two of the disadvantages may be the regeneration process and the process optimization. That's what my research, in fact. So the knowledge gap, so far the research I have found from my literature review, as you can see, and the quick summary is most of the cases, the contact time is too high. So when we are talking about the bulk scale wastewater treatment, which is very bulk volume, we need some kind of a technology which can treat very quickly, like the low retention time. And most of the cases, as you can see, the higher silver concentration have been used, and the performance is like moderately even 400 to 600, depending on the activation or some other doping systems. The knowledge gap I have identified: mitigation of silver pollution by adsorption has not been well explored. And the silver adsorption by biochar has mainly been studied at batch scale. And finally, the reusability is a key factor to determine the potential use of biochar adsorption for industrial application. So as you can see, I identified like 3 research gaps, and today I'm going to share just 1/3 of my study. So that is kind of a one research question, which is the effect of biosphere stock on biochar quality and assessment of their implication for silver removal, where I compare among the 3 biomasses, which was my subject of the presentation. So the methodology. Based on the literature review and availability in Queensland, I have selected algae, waste coffee ground, which is the spent coffee ground, and then sawdust. The reason for this selection, various selection, the algae, based on the literature review, it has like kind of a different composition than other biomasses. For example, it is the seaweed, which has like the mineral residue, which can impact like the remain in the biochar, of course, then can impact the adsorption process. Then the waste coffee ground, especially after the like the coffee brewery or the instant coffee, the waste we generate, which straight away goes to landfilling and creates some like other problem, and it has like more than 1,000 different chemicals. So while the biochar production, maybe some of the chemical residue can help to increase the silver adsorption process. And finally, I also selected sawdust. Sawdust because of the simplicity of the the chemical composition and also the larger surface area. And the idea was to compare among the three, like the different biochar. Then I characterized the biomass by proximate and ultimate analysis, and also I did the pyrolysis gas chromatograph spectrometry, and also thermogravimetric analysis and pyrolysis kinetics and the kinetic and thermodynamic analysis. And finally, I produced the biochar and characterized them by using like the pH, analyzing the pH, surface area, and the functional groups and crystallinity. And I did the pyrolysis with the three different temperature based on the thermogravimetric analysis. We will be talking very soon about those selection. And finally, I tested the silver adsorption performance by using these three biomass, and I also used Taguchi method to design my experiment, because as you can see here, a number of biomasses and other pyrolysis condition I had to vary. So I used Taguchi method to reduce the number of experiment. And finally, I use ICP AES to analyze the remaining silver in the influent and the final treated effluent. And I also use SEM EDS to understand the silver adsorption on the biochar surface. And also I did the XRD analysis to identify the silver species. And also I did the XPS analysis to understand the oxidative state of the silver. Now, the proximate and ultimate analysis data, as you can see here, like these are the three different biomasses. They have like cellulose, hemicellulose, and lignin, and some other residue. And most of the different observation from, as you can see from the ash content, algae has given the maximum ash content, and it's like nearly 20 times than the other two. And like in terms of fixed carbon or the carbon quantity and other, it looks like moderate, because they are the three different source biomasses. Then the Pyrolysis-GCMS analysis, which was done to understand the composition of pyrolysis vapor. So as you can see from here, the Pyrolysis-GCMS data, that the acid compounds, especially the acid compounds, spent coffee has given the maximum acid compound. Similarly, when it comes to phenol, then you see the sawdust has given the highest phenol compound. Then the ketone or aldehydes, as you can see, algae has given the maximum. But when in terms of the phenol, because they are the acidic one, and also the acid compound, which are mainly responsible, these acidic functional groups are responsible for the heavy metal adsorption. So two things we can understand here, that the pyrolysis vapor, as you can see, can show us that these are the remaining chemical compound which may remain in the biochar surface and give us some of the acidic functional group, which will help us to absorb the silver species. And secondly, the high acid value or the high molecular weight compound in the pyrolysis vapor, which we can condense and produce bio-oil, that can offset our pyrolysis cost. Now, I did the thermogravimetric analysis, and as you can see from the thermogravimetric analysis and derivatized thermogravimetric analysis, it has the three different decomposition zones. So when it comes to three different biomass, as you can see, sawdust has given the most simpler decomposition curve. And on the other hand, if you see like the algae, it has like different ups and downs because of its different percentage of lignin, and also the because of the seaweed, it has the kind of a mineral residue inside. But still we can see like 200 to 500 is the most decomposition area of the all the three biomasses. Then I did the thermogravimetric analysis again by using 5 degrees Celsius per minute, 10 degrees Celsius per minute, and also 15 degrees Celsius per minute, like the heating rate, to do the pyrolysis kinetic analysis. As you can see here from the right-hand side, derivatized thermogravimetric analysis, the 10 degree Celsius per minute heating rate has given the maximum decomposition for both coffee and sawdust, except algae. And also, as we can see, when the heating rate is increasing 5 to 10 to 15, then the decomposition curve is shifting to the right side. So which means that before completing the complete decomposition, the temperature is increasing. So that's why the decomposition curve is shifting to the right direction. Now, from the TGA, TGA and DTGA data, I used the OFW and KS method to estimate the activation energy. So as we can see, algae, spent coffee grounds, and sawdust. Coffee has given the required the maximum activation energy, which represent that coffee has kind of a different composition compared to others, and it requires extra energy to decompose. And we have seen also the Pyrolysis-GCMS analysis that it has more like the higher molecular weight compounds than other two biomass. Now, when we compared the two methods, like the OFW and KES method, and we calculated the activated energy, as you can see, the two method has given very similar result and very representative result for 3 different biomasses, regardless of their individual, like the individuality, because they're the different source, so that's why they're different. But the both method has given the similar required activation energy. That can see the compliance of these two methods, which can predict the pyrolysis condition, and we can use those equations to scale up the pyrolysis process. Similarly, I also did the thermodynamic analysis. As you can see, the required enthalpy, Gibbs free energy, and also entropy change, still like coffee is like coffee has shown like more different composition compared to others, and which require extra energy to get the thermodynamic equilibrium while pyrolysis. Now, this is the basic of pyrolysis system, which I did after my kinetic analysis. I used the quartz tube furnace, and on the crucible, I put the biomass and then pass the nitrogen to make the inert environment inside the furnace. Then I collected the biochar, but I just vent the gas, I mean the pyrolysis gas, because my main plan was to use the biochar. I did not collect the bio well or the pyrolysis gas. Sorry. So after producing the biochar, I have characterized the biochar which I produced from 3 different temperature and by using 3 different biomass. So as we know, for adsorption we need like 2 spatial the properties. One is the surface area. The second one is the surface functional group. So as we can see here, the specific surface area or the bed surface area, like the sawdust has given the highest surface area. On the other hand, coffee has given the minimum. As we can see, only 500 degree has given the 41, and algae has given like kind of a moderate surface area. But as I have already said, the surface area is not only the criteria, but also the surface functional group. So to understand the surface functional group, I did the FTIR analysis of all those 3 biomasses and also their biochar fraction. So as you can see from the left-hand corner, the first graph that indicates like the different kind of functional group present in their biomass form. But when I pyrolyzed, then except the 500 degree, like the 700-degree and 900-degree has given the kind of a flat curve, which means all the functional group by the time decomposed and only the carbon residue was remaining. Then the 500-degree, if we compare with 3 biomasses, then we see like the only coffee has the big like the peak, which indicates the sum of the remaining acidic functional groups which we are looking for to increase the silver adsorption. Then I also did the XRD analysis to understand the crystallinity of the biochar matrix. Then I found like the first one is the algae, then second coffee, and third one is the sawdust. And all of them, like the coffee and sawdust, which is more or less the graphitic carbon, there is no sharp peak, but algae, it has some like short sharp peak, which indicates some of the mineral residue from the seaweed. Then I use the Taguchi method to reduce the number of experiments to understand the silver adsorption performance among all those 9 different biochar. So I use like I changed 5 variables like the biomass type, pyrolysis temperature, silver concentration, then RPM, and biochar quantity. And also I checked 3 different layers. And every time I use like 5 hours and 50 ml solution. This is my adsorption process. As you can see, I produced the silver ion solution by using silver nitrate. Then I added like the biochar, and finally I wait for like 5 hours. Then I took some of the solution, then checked by using ICP AES and XRD, SEM, EDS, and XPS. So we will see the result and discuss more about in coming slides. So is the adsorption capacity and percentage of removal, which was the key indicator to determine which biochar is the most effective one or the best one among the 3. So here is the total result of 27 experiments. And as you can see, the middle one, they are the algae, and 99% removal, which is the algae biochar was able to remove the all silver with having different adsorption capacity. But the problem was this is because of the precipitation. As you can see from the middle picture, that when I added biochar in the solution, it produced like white residue and white precipitation. So we can't distinguish between the precipitation effect and the adsorption effect. But when we compare like sawdust and coffee, as you can see, the coffee has given like the 25 milligram per gram adsorption capacity and 99.9% removal. And similarly, sawdust, which was produced at 700 degree, has given the kind of a similar adsorption capacity and the removal rate. But when we compare with these two, coffee biochar will require less energy to produce this biochar and getting the same adsorption performance. But sawdust, we need like 700 degree, which requires higher energy. So this observation can help us to decide that coffee biomass is the best biomass among the 3 biomasses for silver adsorption. So to understand the silver adsorption, like the property on the surface, I did the ACM ADS analysis of the before adsorption biochar of 500 degree coffee. And as you can see here, the 2 pictures, this is a little bit different than other lignocellulosic biomass derived biochar, where we see like kind of a honeycomb shape and very even micropores, but this is kind of a scattered surface, as you can see. And the if we see the like the particle size was too small, like more than 50% is like kind of a 184 micrometer. Then I take the SCM-ADS picture of like the silver adsorbed biochar. As you can see from the second line of the picture, the middle one, red one, is the backscattered image of the biochar surface. And the left and right side, as you can see, the white dots, which are basically the metallic silver, which is identified the metallic silver, and the EDS mapping tell us the like the ratio of silver adsorption on top of the biochar surface. But these white dots are really silver or not. To identify this, I did the XRD analysis again. So the before adsorption, as you can see, the left side, and the after adsorption, you can see the right side, the sharp peaks, which is indicating the region is indicating the presence of metallic silver. But still, we don't know which form of silver is present in this biochar. I mean, I use silver nitrate, I mean silver ion, but what was the chemical transformation? So to understand these things, I did the XPS analysis, like before adsorption and after adsorption. The left hand side is the before adsorption biochar, and the right hand side is the after adsorption. So the summary of the result, as you can see, in terms of silver ion, before adsorption, there was no silver because this is the virgin biochar. Then when I use like the after the adsorption, we can see there were like silver ion as well as silver like the nanoparticle, silver 0 oxidative state, and also silver 2+ oxidative state, which indicates we use silver ion, but it transforms into silver like the metallic silver, which is 0 oxidative state, and also like the double oxidative form. So we can clearly see the chemical transformation of silver ion. Similarly, the carbon concentration, if you see the graphite carbon and aliphatic carbon was 0 at the beginning, but after adsorption, it also we can see some of the percentage, like the atomic percentage 2.6.2, which indicates the the chemical changes of biochar after the adsorption. So now we can see why sawdust has given the highest surface area, but given the low adsorption capacity, because the adsorption capacity was dominant by the chemical transformation or the surface functional groups. And the conclusion of my study is algae had very high ash content, which is not feasible to produce like the biochar. Then the coffee biochar had the more functional group, which is helpful for silver removal. Then the 10 degree per minute heating rate and 500 degree is the optimal pyrolysis condition for coffee biochar production. And coffee biochar showed the higher silver adsorption, definitely among the three. And finally, the functional group determined the total adsorption. And SCGs also, I mean, the coffee is also promising biomass for other byproduct recovery to offset the pyrolysis cost. So this is all about my first, I mean, the 1/3 of my research. And thank you for listening. If you have any question, please feel free to ask. Thank you.
Thank you, Anwarul, for your presentation. As always, really nice to listen to your work. You got a question about your sawdust. Do you know the species, the wood species you used, or where the sawdust came from? Was it softwood, hardwood?
Yeah, it's a hardwood, Australian pine.
Australian pine.
Yeah.
And I would be really interested in the recycling regeneration, but I guess that's not part of your presentation today.
No, I did the regeneration. That is my 3rd chapter. As I told, like I had the 3 questions. So one is to identify the best biomass. That is my 1 chapter. The other one is I did the batch scale study. And then the 3rd one, I did the continuous study. I designed a pilot and I ran the pilot. And finally, I was able to do the 3 cycle of regeneration by losing kind of a 14% efficiency.
Okay, sounds quite promising. For the algae biochar, so you said it started with 23% ash content.
Yes,
and I saw you got a biochar yield of 27%. So yeah, does that mean you only have like 4% carbon left?
Yes, okay. Because it decomposed completely.
Okay, yeah. So how did it look? The
So I mean, it still is kind of a not the usual biochar like the black one. It's kind of a ash, the
Okay.
you know, the gray color. So
Yeah.
the yeah.
Okay.